Local, resting, and action potentials. Resting membrane potential. The cell has a resting membrane potential because electrolytes are distributed differently between the extracellular fluid on the outside of the plasma membrane and the intracellular fluid on the inside. Sodium leaks into the cell and potassium leaks out, but the potassium-sodium pump continually compensates for the leakage. It pumps 3 positive sodium out of the cell for every 2 positive potassium that brings in. By removing more cations from the cell than it brings in, it contributes about -3 millivolts to the resting membrane potential. The resting membrane potential of -70 millivolts is the net effect of all these ion movements. Potassium diffusion out of the cell, positive sodium diffusion in, and the positive sodium with the positive potassium pump continually offsetting this ion leakage. The resting membrane potential results from the combined effect of 3 points. 1, the diffusion of ions down their concentration gradients through the membrane. 2, selective permeability of the membrane, allowing some ions to pass more easily than others. 3, the electrical attraction of cations and anions to each other. Local potentials. The response starts at a dendrite, spreads through the cell body, travels down the axon, and ends at the axon terminal. The incoming positive sodium diffuses for short distances along the inside of the plasma membrane, creating a wave of depolarization that spreads out from the point of stimulation. This short-range change in voltage is called a local potential. 4 characteristics of local potentials are: 1, local potentials are graded, meaning they vary in magnitude, voltage according to the strength of the stimulus. 2, local potentials are decremental, meaning they get weaker as they spread from the point of origin. 3, local potentials are reversible, meaning that if stimulation ceases, cation diffusion out of the cell quickly returns the membrane voltage to its resting potential. 4, local potentials can be either excitatory or inhibitory. So far, we have considered only excitatory local potentials, which depolarize a cell and make a neuron more likely to produce an action potential. Action potentials. An action potential is produced by voltage-gated ion channels in the plasma membrane. The neuron reaches its threshold membrane potential. A neuron membrane with a -70 millivolt, which is generally around -55 millivolt. When the threshold is reached, several sodium channels open, which allows positively charged sodium ions into the cells. This causes depolarization of the neuron. As the membrane potential reaches 0, it then becomes positive. This is the rising phase of potential. The influx of positive ions creates electrical signals known as the action potential, which then travels down the neuron. Then it reaches its peak, where the sodium channels close and the potassium channels open, allowing potassium to flow out of the cell. This loss of potassium ions causes repolarization. Then the neuron returns to resting membrane potential, but actually overpasses and the cell becomes hyperpolarized. Eventually, the potassium channels close and the membrane returns to resting potential and ready to be activated again.